right, good morning and welcome everyone. Thank you so much for joining our second RID-HIV CAB quarterly webinar. And we're delighted today to have Dr. Paula Cannon, one of our primary investigators from the collaboratory, talk about her exciting work in gene therapy. So welcome, Dr. Cannon.
Thanks, Jeff. Okay, let's get started. Oh, fail already? Nope, got it. Okay, all right. So today I'm going to be talking about one of the projects I'm doing for RID-HIV. Here's my title, Reprogramming B cells to make better antibodies against HIV. And we'll go over what antibodies do and why we might need better antibodies and one of the ways we think we might be able to do it. Okay, so let's just start off with a recap. You guys probably know this, but here's my take on what HIV's sneaky tricks are and why it's been so difficult to both make a vaccine against this virus and to cure people who are infected. Okay, so the first thing is that HIV inserts its genetic material, we call it its genome, into any cell that it infects, and therefore it has the potential to become permanently a part of the DNA of that cell. So on my little cartoon over here, hold on a minute, let me just get a laser pointer. It's my new thing. My little cartoon over here, I've got HIV entering a cell, and here's its genetic material. It's driving, it provides the instructions to make new viruses. And if we homed in on the nucleus of that cell, you'd see it's full of human DNA in our chromosomes. And here's the HIV virus that's just randomly inserted itself. So basically, it becomes the equivalent of an infectious genetic disease. The next thing to note is that, hold on a second, I just need to try and move the little bar at the top because I can't read my titles. Stupid trick. Let's see, put it down there. Right. Okay. Sometimes those HIV viruses, after they've integrated into the genome, instead of actively producing new viruses, as in this case, they become latent. The genome switches off, but the genome is still there. So this is like a genetic parasite in the cell now, and we call these cells latently infected. And because they aren't making new viruses, they don't look any different than uninfected cells, and they're invisible to the immune system, and therefore they can persist. And I think everybody understands the consequences of that, which mean that, okay, good, it's working. That this is showing what happens, that if you're on antiretroviral therapy and you stop taking antiretroviral therapy, you get viral rebound, because if even one of those latently infected cells reactivates, it's kind of like a spark restarting a forest fire you think had gone down. And this reservoir of latently infected cells is the reason why antiretroviral therapy can suppress HIV levels really well, but can't currently cure people. Okay, here's the third sneaky trick of HIV. It mutates like nothing else on the planet. And as a result, it can evolve away from even the best efforts of our immune system. So just a little bit of immunology shown here. On the left is one branch of our immune system, the CD8 T cells. CD8 T cells recognize and kill HIV-infected T cells, because those HIV-infected cells, for example, will be showing bits of the HIV spike protein, the HIV-M protein, these orange blobs on the surface that can be recognized by a sort of an antibody-like structure on the surface of a CD8 T cell that we call the T cell receptor, or TCR. And then the other main branch of the immune system comes from our B cells, which make antibodies that can stick to and neutralize HIV. And interestingly, the B cells also, on their cell surface, they express a sort of an anchored version of the antibody that we call the B cell receptor. And so a really central aspect of immunity is that although we have, millions and billions of different CD8 T cells and B cells in our body, they all show on their surface a different TCR or a different BCR. And it's only when those particular TCRs and BCRs recognize an antigen such as the HIV envelope protein that they can specifically bind to, that the cells themselves become activated and start doing their job. So this is sort of a basic premise of immunology and how different pathogens and different antigens can recognize and activate their own unique T cells and B cells. Okay, and then, so despite these best efforts, you know, HIV mutates a heck of a lot. It's got a very error-prone replication system, and as a result, it can easily brush off even the best antibodies that we can make. And here's just showing it in color. The purple HIV, the body responds to make purple antibodies that fit really well. But even if just one HIV now evolves into the red form that can no longer be recognized by the purple antibodies, that puts pressure on the body to make a new type of antibody. Let's call it red antibodies. And the virus, like a chess match, just fights back and mutates again. So in HIV infection, people constantly make antibodies that should work against the virus, but the virus responds by mutating away from those antibodies. Okay, and then finally, the final sneaky trick is HIV, as everybody knows, infects CD4 T cells. And this is really critical because CD4 T cells are like the conductor of the orchestra of the immune system. Because HIV infects those T cells, it blocks their function. And sometimes we call these CD4 T cells helper T cells because they help the rest of the immune system do their job. And in particular, they help both B cells and CD8 T cells. So if HIV comes along and infects a CD4 helper T cell and then wipes out that T cell, then what happens is we lose this communication with the rest of the immune system and we have decreased immunity. Okay, so that's by way of introduction to RID-HIV, which I think I got this right, stands for Reversing Immune Dysfunction to Cure HIV. And just to quickly introduce you to everybody on it, because we're a good-looking group. Here's the three principal investigators, Sumit, who you guys have heard from, Rafiq, and myself. Here's Jeff as our community engagement coordinator. We got an amazing scientific advisory board, but the bulk of what we're doing is down here by this amazing group of scientists, and of course all the scientists in their lab, and we are all laser focused on understanding and reversing the immune dysfunction that is a block to curing HIV. So here's kind of how I think of what RID-HIV is doing. First of all, we have lots of projects wanting to understand exactly how the immune system is changed by HIV infection, kind of know the enemy. Then we have projects that want to enhance these different components of the immune response. And finally, we also have projects that are aimed at reducing this problematic reservoir of latent HIV genomes. So my little piece is in the middle section about thinking of different ways that we could enhance the various components of the immune response. Okay, so another little bit of background I want to tell you about that in non-HIV fields, especially cancer, we're seeing amazing progress in people figuring out ways that they could engineer CD8 T cells. So remember, CD8 T cells are the ones that express the T cell receptor on the surface that allows them to recognize an HIV-infected T cell, and they can kill that cell. So as I said, we're seeing amazing progress in cancer. Okay, a bit of a busy slide, so I'm going to talk you through it. So here's on the left is what happens naturally. A tumor cell expresses on its surface through a sort of a carrier molecule called MHC class 1, more detail than you need, a little bit of that tumor cell that kind of tells the immune system that that tumor cell is basically foreign, it's mutated. And the T cell receptor on the surface of the CD8 T cell locks into and recognizes that and kills the cell. So this cartoon here is showing natural recognition of a cancer cell by the T cell receptor. But one of the problems with cancer, and the reason we don't all cure ourselves of cancer, is sometimes the T cell receptors don't do this job well. They don't recognize the tumor cell, or there's other reasons why they don't work. And so there's this whole technology called CAR T cells. And I've just put over here some of the many CAR T cell therapy companies that have developed in the last decade. And that tells you it's a hot topic. So if you're looking for some investment opportunities. So what CAR T cells do is the T cells are taken out of the body and they are engineered to express an artificial T cell receptor that we call a CAR, or a chimeric antigen receptor. And as a result, these CAR T cells can now recognize a target on the tumor cell, home into that cancer. And when they work, they wipe out the cancer. So there's been some extraordinary successes in cancer. So for HIV, some groups are trying to think of ways that we can create CARs that recognize, instead of a cancer cell, an HIV infected cell. So for example, again, here's my cartoon version of what you just saw. Here's the natural recognition of a HIV infected cell. It's expressing the HIV ENV on its cell surface, which is a target for recognition by the natural T cell receptor, or that can be replaced by an artificial T cell receptor, by a CAR. And some of the opportunities for making CARs include adding parts of antibodies that naturally recognize the HIV envelope protein, or even taking the natural receptor of the HIV protein, which is the CD4 molecule, and taking like the N little blob of CD4 that HIV ENV binds to, and using that as a CAR. Okay, so I'm not going to actually be talking about CARs. So all that background was kind of to set you guys up to thinking, to sort of showing you the journey I went on. Because as I started reading about CARs and TCRs and CD8 T cells, it occurred to me that B cells are very analogous, that the B cell receptor kind of does a similar job as the T cell receptor. It recognizes and binds to its specific target antigen. And as a result, the B cell then differentiates, expands, gets activated. And in the case of the B cell, starts to spew out antibodies that could kill that virus. And so I wondered if we could think about engineering the B cell receptor and the antibodies that the cell produces. So that's where we start, with this idea from a few years ago now that I had of, can we use gene therapy to make better anti-HIV antibodies? Okay, it's a very long introduction, and it's going to get worse because I still have more background to give you guys. All right, so here's a busy slide about broadly neutralizing antibodies, or BNAbs, that you guys probably know about, but I'm going to go through their main features. So again, as I've already said, HIV is very mutatable, and its cell surface envelope protein, it's also called GP120. It's very analogous to the spike protein in the coronavirus. That also easily mutates and changes. And so if there are antibodies that are recognizing the GP120 protein, little mutations can easily throw off the antibody recognition. However, rare broadly neutralizing antibodies have been identified that are hard to mutate against. They neutralize diverse strains of HIV, and there are many different strains of HIV, especially when we look globally across the world. And these BNAbs, not only can they neutralize a broad range of diverse strains, they also then resist this drive to mutate and escape from antibodies that HIV has. And as a result, people can make these BNAbs. They can make them as what we call recombinant proteins, like monoclonal antibodies, and they can be injected into somebody. And they're starting to show promise in clinical trials as a sort of a biological drug, especially when more than one BNAb is used. So BNAbs are great, and it would be fantastic if we could think of how to vaccinate people, both HIV infected and uninfected, as a preventive measure to make BNAbs. But unfortunately, all current vaccination strategies have failed to create BNAbs. When people get vaccinated with the HIV ENV protein or some other derivative, they make lots of antibodies, but they are these regular antibodies that HIV can mutate away from, and not these rare, and generally not these rare broadly neutralizing antibodies. So I think there are people in some of the other collaboratories who are working on this idea of, can we figure out a special vaccination strategy to create BNAbs, but that's not what I work on either. Okay, so sorry guys, it's just such good coffee and I can smell it while I'm talking. So final piece of background is that Antibodies are kind of here to stay and are massively exploding. Antibody therapies are now used for many chronic disease conditions. And so in my little cartoon guy here, he's been injected with these monoclonal antibodies. And they're great when they work, but they do need re-administration, perhaps monthly, and they come at a high cost. And this little table down here kind of freaked me out when I first saw it. This is from like 6 years ago, but this is the annual cost of some monoclonal antibodies that are used. And you'll see, especially in the cancer area, oncology, hematology, you see some of these. I mean, look at these. These are eye-watering costs of these antibodies. So they're great, but they're kind of highly specialized at the moment. Because of this expense and inconvenience, people are definitely excited in the possibility of using gene therapy as a way to introduce, for example, a gene therapy vector into the body, which will deliver the genetic material to make those bNAbs in vivo as a sort of a factory inside the body. And typically, people are injecting these antibodies into either liver or muscle. So the antibodies are expressed from non-natural cells, from cells that are not B cells, such as muscle and liver. Okay, oh, no. Okay, this is a busy slide. Let me walk you through this. So I just said that antibodies can be expressed from non-natural cells. But antibodies made from natural B cells have distinct advantages. So I'm going to walk you through this little wheel. So first of all, B cells have something called memory. We all understand that because we've all been boosted against COVID. And what that means is that B cells, so here's my B cells with their specific B cell receptor on their surface. And this could be a sort of a B cell receptor that recognizes COVID. If those B cells meet their antigen, either because they get reinfected or they get vaccinated, they will boost, they grow, they divide, they differentiate. So we can keep expanding the number of B cells that are specific for our antigen. And as a gene therapist, I kind of love this aspect because it means I could actually be a pretty lousy gene therapist and only engineer a few B cells, but nature will then sort of take over and amplify what I wasn't able to do. The next thing to kind of point out is over here. I call them systemic antibodies. Once B cells differentiate into the antibody secreting cell, and you'll see the difference. Here I've got B cells with the antibodies stuck on the cell surface, waiting to meet their antigen as the B cell receptor. Once they've been triggered by that interaction, they then become what we call plasma cells. And these spew out antibodies, and they are long-term, years, decades, spewing out these antibodies. And so these cells are kind of natural factories in the body. And so there's a lot of excitement about, well, if we could tap into that, we have a way to think about making not just antibodies, but maybe other secreted factors in the body in a very efficient way. Let's go down here to the bottom left. Another thing that we're starting to understand about B cells is they're not just swimming around in the blood. They traffic to specific areas in the body and especially to lymph nodes. So we can think about ways that our B cells may be able to go to places where HIV infection is concentrated and deliver the antibodies specifically there, not just, it's great to have them swimming around in your blood as well, but that targeted local delivery might be cool. And then finally over here, antibody evolution. This is this weird and very cool trick that antibodies do. In that once an antibody has been made, the body isn't kind of satisfied with that. The antibody genes themselves undergo a process of tweaking and mutation that can then be acted on to select for better and better antibodies. And so in theory, if a virus change, from blue to orange, if some of the antibodies have also changed to be a better fit to that new virus, then those antibodies and the B cells that make them will be selected for and become the sort of the dominant antibody producing cells. So there's really just a lot of amazing biology behind antibody production from natural B cells that you don't get if you're just expressing an antibody from a liver cell. So when I first understood this, I got quite excited about this. But here's the tricky thing. Many of these properties of B cells are a consequence of where the antibody gene is made, because it's expressed from a gene called the immunoglobulin locus. And therefore, if you want to have an antibody that recapitulates these features of B cells, you have to think about being able to express that antibody from the natural immunoglobulin locus. Okay. Oops. So our basic idea was we knew that CRISPR-Cas9 gene editing, and I'll talk about that on the next slide, can give us a capability to very precisely insert new genetic material at precise places in the genome. And so our question was, can we use CRISPR-Cas9 gene editing to express our antibodies, in this case, our bNAbs, from the natural immunoglobulin locus? Bit of background about CRISPR-Cas9. You really don't need to understand all this, but here's a cartoon of it. And basically, CRISPR-Cas9 cuts DNA at a site, any site, that's determined by its partner CRISPR. It's called a guide RNA, and it drags the Cas9 protein, which is this pinkish blob, to a specific region on the DNA. And then that DNA is cut, so we get a specific DNA break at our target site. And then as a result of that, we can then use the processes of the natural processes of how the cell repairs a DNA break to force it to insert our new genetic material, in this case, the immunoglobulin locus. And so what happens is this DNA break is repaired by patching from what we call a DNA homology donor that we introduce into the cell. It's a very complicated process, so I'm not going to go through it on this slide. But the end result is we can get insertion. Here, my green arrow is supposed to be my antibody. In theory, we can insert our antibodies into the immunoglobulin locus. So that's the sort of enabling technology. Okay, so when we started on this project, I thought, okay, this is a really cool idea. Great, let me put a graduate student on it. This will work straight away. And then I started reading about the immunoglobulin locus. That's what IG stands for. And I started to get a bit scared about the project we'd embarked on. It's a couple of problems. First of all, antibodies are complicated. Okay, so let's do a bit of antibody biology here. This is my cartoon of an antibody, and they consist of 4 chains, 2 heavy chains, these are the long ones, and then 2 light chain partners. And then, as I've already explained to you, they can be both a secreted antibody as well as anchored onto the surface of the cell by having this additional little gray blob. It's like a membrane anchor, and this forms the matched B cell receptor. And then if that wasn't bad enough, the immunoglobulin genes are also complex because there are 3 different genes you'd have to think about engineering. There's one gene that makes the heavy chain, and there's 2 genes, kappa and lambda, that make the light chains. And then in addition, all antibody sequences are different. That's why they work. And in particular, these head regions, the red and the brown components, one comes from the heavy chain, one comes from the light chain, and they sort of bind together to create the sort of unique variable head of the antibody that recognizes every different antigen. And remember, they undergo mutation. So what this means is that if I took a million, 1,000 B cells out of an individual and I wanted to use CRISPR-Cas engineering to insert antibodies, the problem I've got is that 1,000 B cells will give me 1,000 slightly different sequences at the immunoglobulin locus. And because CRISPR-Cas9 gene editing is all about bringing the CRISPR-Cas9 machinery to a specific sequence, having a locus that is hypervariable and is different is immediately a problem. And then finally, we have to worry about what I call mismatches. That if I go into a cell that already is, that is already making an its own antibody with random heavy chains and light chains, and I introduce foreign ones, then there's nothing to stop those mismatching and having, you know, kind of, if you like, different green and yellow combinations. And because the heads then are going to be made of different combinations of heavy and light chains, I'm going to start creating completely random antibodies that could create problems, sort of start to attack something in the body. So lots of problems to think about. Oops. Great. Okay. Here's what rescued us, the camel. It's kind of a picture, isn't it? Yeah. So let me explain to you why camels have come to our rescue. Okay. So camels make a special group of antibody that we call heavy chain only antibodies. And if you just look over here, here's the diagram of the typical antibody I've already shown you. And here's what the heavy chain only antibodies look like. So you'll see they only have one chain. It's a heavy chain. They don't have the light chain partner. And in addition, if you look carefully, you'll see that although these two regions that are called CH2 and CH3 are present, this region up here called CH1 in the heavy chain is missing. So these heavy chain only antibodies naturally occur in camels and llamas and alpacas, and they occur alongside their conventional antibodies. The head group, which is called VHH, or it's also called nanobodies, it's only made of one component from the heavy chain, so it's very sort of compact and stable and small, and therefore there's a lot of excitement about using these as recombinant injected antibodies in people. And then, as I've already told you, this CH1 domain is missing. And this is critical because CH1 is the region that recruits and binds to the light chain partner. So by not having a CH1 region, these heavy chain only antibodies don't need a light chain partner. And in fact, they won't bind to a light chain partner. So remember the mismatching I was worried about on the previous slide? At least some of that potential mismatching between my heavy chain only antibody and the light chain that's been made naturally in the cell, at least that is prevented in this situation. Okay. So this is our general idea that by mimicking the idea of these heavy chain only antibodies, we could use a simplified CRISPR-Cas9 gene editing approach that only needed us to go in and edit the heavy chain gene. We can ignore the light chain genes. And because the head part of our antibody, this nanobody part in orange, comprises only one kind of protein domain, it opens up the possibility for us to use both antibody and non-antibody components when we do this engineering. And I'll explain about that at the end of the talk. Okay. So I've given you an awful lot of background and it's quite detailed. So I hope you've at least followed some of it up to this stage, because it's going to get worse from here on in, because I'm going to start showing you data, but it's all right. You can just look at the titles. Oh, okay. So the next couple of slides are pretty hardcore, but I put them in for the people who are interested. So this is an overview of the CRISPR-Cas9 gene editing strategy we developed. So first of all, this is a cartoon of the heavy chain gene. There's a promoter which drives production of these different components, VH, CH1, 2, and 3, with and without the membrane. And when all of these different components get made, we end up with two different products. One that just has the red and blue stitched together, and this becomes a secreted antibody, or at least the heavy chain part of the secreted antibody. You see my color scheme with the red and blue. And then alternatively, some of these also have the membrane anchor, the gray blob attached. And so they form the heavy chain component of the B cell receptor. So when the products of this gene naturally are made and partner with their L chains, we get both B cell receptors and antibodies. So that's how the natural antibodies are made. So with our gene editing strategy, we want to use CRISPR-Cas9 editing to come in between the CH1 and CH2 regions and insert a new promoter to drive expression starting at this point, and our new antibody head group, which is this orange box. And once this is inserted into this region, this is what it looks like. So now, instead of this promoter governing which gene is made, instead, this inserted promoter works and it produces these products, the orange and blue spliced together with and without the extra gray membrane. And these products, these are heavy chain only components. These comprise these heavy chain only antibodies and the matched B cell receptor. So that's the basic gene editing strategy. At least that was the idea we had. So of course, does it work? So the project that we did, first of all, we thought, well, let's select a test head, the orange box that we're going to insert that will recognize the HIV envelope, HIV GP120. Then we had to do some work to optimize the CRISPR-Cas9 gene editing tools. And then finally, we had to test if the edited antibodies expressed from the immunoglobulin locus in B cells could actually make it out of the cell and work to neutralize HIV. Okay, so I'm going to introduce you to the tool we used. This is J3. It's a broadly neutralizing antibody from llamas. Okay, this is kind of, remember, this is the what the heavy chain only llama antibodies look like. And we took the head of this llama antibody to make our cassette to be inserted. And over on the right here is data I took from this paper, which is just showing that the J3 VHH domain or nanobody head is indeed a broadly neutralizing antibody. For those of you who look at this data, that these are all different subtypes of HIV, subtype A, B, C, and this is just showing for different viruses in those subtypes that were tested with this antibody, how good the antibody was. When it's red, it means it was really powerful. Yellow and green, less so. But you see there's a great breadth across these different subtypes of HIV. So J3 is a pretty nice llama heavy chain only antibody that potentially, you could inject into people and expect it to have powerful anti-HIV properties. But what we want to do is we want to use J3's head and engineer it into human B cells. So yes, putting a little bit of llama into people. Okay, so probably more detail than you need, but I just want to point out that this took an awful long time to do and was really hard work for my students. And sometimes when I talk about this work at CRISPR-Cas9 gene editing meetings, basically this is like 25 slides, okay? So I'm not gonna inflict that on you today, but the simple concepts are, we made lots of different CRISPR-Cas9s that could make DNA breaks at slightly different places in the immunoglobulin locus, for those of you who remember, between the CH1 and CH2 regions. And then we tested them all, and we were looking for CRISPR-Cas9s that had really high on-target activity, which these are the black bars shown here. This was the locus we were going after, but ignored highly related genes that we didn't want to target. So you'll see, for example, this one, we call it SGO3. It gave really high levels on target, but we didn't use it because it also gave high levels off target, which means that our antibody could be inserted at the wrong place. And instead, we liked number 5, which had pretty decent levels on target and gave us no gene editing at other sites. Okay, so the next thing we did was we asked whether or not these antibodies worked. So we asked whether CRISPR-Cas9 J3 edited human B cells actually make functional anti-HIV heavy chain antibodies. Bit of a mouthful, that title, isn't it? So the first question we had was, well, can we detect J3 on the cell surface as a BCR? And this experiment is just showing that the engineered cells, but not control non-engineered cells, recognize the HIV-ENV protein GP120. So yes to that question. The next thing we did, again, this is probably more detail, was we said, if we take those B cells and we differentiate them, we treat them with chemicals that persuade them to be not just a cell that's expressing the BCR on the surface, but to change into these plasma cells, these factories that start to secrete the antibody. If we do that, do we see a switch from the membrane-anchored BCR to the secreted antibody? And that's what this assay up here shows. And the answer is yes, over time. By day 8, we had hardly any of the membrane M form, and everything was the secreted S form. And then finally, and most importantly, we did an HIV neutralization assay. We took the cell culture from the edited B cells, which would contain, if it was working, our secreted antibodies. And we added it, we mixed it with HIV virus, and then we looked at the infectivity of that virus. And you'll see that as we added more of the supernatant, the antibodies, you'll see that HIV infection was inhibited, whereas the culture from our control cells had no effect. And then finally, this is kind of a cool thing. Remember, I talked to you about how antibodies, B cells, mutate their antibodies a little bit. So there's the potential for evolution and the potential for the body to select for better and better or more specific antibodies. We did an experiment in cell culture where we sort of mimicked that. Basically, we kept our engineered B cells in culture over 8, 16, 24 weeks. And at 3 different points, we sort of selected for the group of B cells that stuck to HIV GP120 with more affinity. You'll see you see over time we get more and more in this little box. And then when we took the antibodies that those selected cells made, we saw that again over time, this is just showing you that their ability to bind to GP120 increased. So at least in vitro, what we've proved here is that our antibody cassette that we've inserted at the authentic immunoglobulin locus is able to act like a natural antibody. It undergoes mutation. And if that mutation was selected for, we've done it in vitro in this artificial way, but the body will also do that. Then there's a potential that we can get even better, sort of tighter binding antibodies. Okay. So, and that's just showing you what we did. I'm going to skip that because it's just repeating what I've said. All right. So the last little thing I want to tell you in the last few minutes is that one of the things I like about this antibody engineering is, in theory, we're not limited to using natural antibody components. So I've shown you a proof of principle where we took a llama nanobody, a llama heavy chain only antibody head, and we use that to insert into the human immunoglobulin locus. But that's kind of a natural antibody component. So let me walk you through the sort of what I think is the flexibility of this system. So here's just my cartoon with the head and then what I call the stalk of these heavy chain only antibodies. So first of all, in the head, we can put in antibody components. I've shown you we can use camel and llama nanobodies, but we can also use other antibody components. You may have heard of single chain antibodies, for example, and we can engineer those there. But we can also use completely non-antibody components. And when we're thinking about something that could bind to and recognize the HIV envelope protein, the non-antibody component that jumps out is the CD4 protein, because we know that that's what the ENV naturally interacts with. And if you remember way back at the beginning of my talk, when I was telling you about the CAR T cells, they are using CD4 domains. So we're excited to use those. And we can also, improve on nature. We can actually, instead of just having one head, we can link together 2, 3, or 4 heads as well. And we can make sort of, daisy chained completely artificial, but I think very powerful antibodies. Not being content with messing around with the head of the antibody, we can also start to think about the stalk, because the stalk of the antibody, sometimes it's also called the Fc region, that's not just a display pedestal. Itself, it has important properties, and we can introduce mutations into the stalk that can enhance our antibody properties, such as increasing its half-life and stability in the body, and enhancing other killing mechanisms. And there's a real area of excitement at the moment around the idea of using a different cell of the immune system that I haven't talked about, called natural killer cells, which basically if antibodies are stuck on the surface of a cell, natural killer cells recognize the stalks of the antibodies sticking out and can come in and kill those cells as well. And we can change the stalk to make it recruit the natural killer cells even more efficiently. So that's kind of, tapping into yet another component of the immune system. And then beyond antibodies, and indeed beyond HIV, we can think about just use to stop the recovery. Great. Okay. So beyond antibodies, we can think about using this system to actually express things that aren't antibodies, things that are proteins that could be missing in certain genetic diseases. Think about the clotting factors, for example, in hemophilia. And we can use the power of the B cells for long-term in vivo production. Okay. So just quickly showing you what these alternate heads look like. This is my cartoon of where we've inserted our cassette. We've been using, in my example, with the J3 nanobody component, but we can also put in single chain antibodies, and we can derive those from other well-known bNAbs. You may have heard of PGT121, for example. So we've done that. We can also make these daisy chained antibodies. We've put in multiple different nanobodies, and then we can also put in something like the CD4 receptors. This is work that's ongoing in my lab. These are all targeted to HIV, and we're trying to figure out which is going to be the best approach. And then just a cartoon showing you how we can modify the stalk, the Fc stalk. Again, this is the cartoon of the natural antibody locus that makes natural H heavy plus light chain antibodies. This is the strategy I showed you, where we insert our head downstream of the CH1 cassette, and this is what we end up with, where the variable region is replaced. But we are also now showing that we can go into other regions. And for example, if we went into this region here with our CRISPR-Cas9 editing and inserted not just a head, but a head plus the first part of the stalk, we can end up with a heavy chain antibody that looks like this. And this green blob, this part of the stalk, we can put in anything we want and include mutations to enhance the function of the antibody. Okay, so in summary, we've copied the design of the heavy chain only antibodies that are made by camels to facilitate us engineering human antibody genes in human B cells. We've shown that our gene-edited B cells express our desired custom cell surface antibodies, the so-called BCRs, that respond, in our case, to interaction with the HIV-M GP120 protein and can be differentiated to secrete antibodies that are functional and neutralize HIV. Excitingly, our engineered antibodies also undergo mutation that could be acted on to improve their properties. And in theory, we could insert any protein domain, not just natural antibody components, allowing for complete customization. And I'll just finish with where I think we can also take this, because as in many cases, things that get started because of funding for HIV can go on to change the world. We all know that, don't we? So here's my pitch for where this could go beyond HIV. I think this is exciting to think about a way to make antibodies for other hard to vaccinate diseases. Of course, HIV, I think, is the biggest challenge in the world today, of a disease where we know we don't have a vaccine. But there are other diseases that are also hard for us to naturally vaccinate against. In addition, you can think about using these to make antibodies in immune-compromised individuals. And a great example there that comes to mind is for COVID. You know, we all know that a significant portion of the population, even when they get vaccinated, don't make antibodies that in sufficient levels against COVID. So we can think about putting anti-spike antibodies in B cells of these individuals. We can also use this technology to express antibodies that normally would never be made. And so these are what we call anti-self antibodies, antibodies that actually recognize a natural protein in the body, such as something that's on the surface of a cancer cell. They're self, if you like, so we can't vaccinate to make them, but we can artificially put them in. And then, as I mentioned also, we can think about beyond antibodies secreting factors that are missing in different genetic diseases. And with that, I'll just recognize the members of my lab who are involved in this, especially Jeff Rogers, a senior postdoc in the group, and thank my funding, which is mostly from RID-HIV for this project, but I've also managed to sneak a little bit of funding for another collaboratory, the I4C Collaboratory. Okay, and be happy to take any questions if we still got time. Yeah, I see we still have a few minutes.
Yeah, we still have about 10 minutes. So thank you, Paula. That was terrific.
So it was pretty detailed. I apologize it was quite so detailed, but I thought, you know what, you guys can handle this.
Well, and you did an excellent job as always of walking us through it. So it's all about breaking it down.
Yeah. So
questions for Dr. Cannon?
Well, a basic question from me here. Like assuming, well, nothing goes perfectly in research, but assuming that all you see ahead of you works, how long do you think it will take you to go into just a pilot in humans or even in non-human animals?
Yeah, so one of the exciting things about the Delaney collaboratories, Nelson, is both RID-HIV and I4C are very focused on testing things in non-human primates as soon as possible. So the process we're going through is when we think about switching to testing things first in rhesus macaques, as a gene editor, I have to say the first thing I go is, oh no, because because CRISPR-Cas9 is so specific, it also means that everything we've done and all the tools we've developed work really well on the human immunoglobulin locus, and we'll completely ignore the monkey one. So we're back at sort of step one, developing the tools that will work in the primates. That's okay. We've already done it once. It's not going to take us 5 years. You know, that's well underway. So in both of those grants, we are focused on recapitulating everything we've done in rhesus macaque B cells. So we're getting the macaque B cells, we're learning how to culture them, and we're developing the tools. So my hope is that towards the end of year 2, or possibly year 3, we've just ended year 1 of the collaboratories, we'll be ready to go and engineer macaque B cells and put those B cells into some macaques. And then what we would do is we would follow up with a sort of a vaccination, because again, remember, we're bad gene therapists. We may only engineer a few cells, but if we then come in and vaccinate the macaques with GP120, the small number of B cells we've edited should expand and explode, and we can then test whether or not we are getting our heavy chain only antibodies in the blood of those monkeys. And I think if we saw that, we then challenge the monkeys with HIV in one project and SIV in one project, which has got an HIV-ENV and SIV in the other. So yeah, both projects are different, which I think is exciting because I don't know which one will work. And then we can really start to think about expanding that massively to start to do the safety, considerations we would have to do. So I think, within the 5-year lifespan of both of these collaboratories, we should actually get, if it's going to work, I think it will work quickly and easily. I shouldn't say that, should I? But it'll either, if it's going to work, and sometimes things do, sometimes things actually just work. I think if it follows all the biology of B cells, I think it should work quite quickly. And then, towards the end of the 5 years, that was when I would anticipate we would start to talk to the FDA and say, hey, we've got this completely new, technology. What sort of safety studies should we do? The other thing that's quite exciting, Nelson, is that, I'm not the only person who had this idea. And there are a few companies now and some academic groups who are also working on engineering B cells to make custom antibodies. They're not using the heavy chain only approach we are, which, I just think that's the best idea ever. They're using, other approaches. But the good news is we will all share information and figure this out together. And again, there are people who are interested in doing this for HIV, and there are people who are interested in doing this for other diseases, including cancer. So just as CAR T cells kind of spent a long time in the lag phase and now have exploded, I think engineered B cells, we're in the lag phase, but I think, there will be an explosion in this as well.
Thank you. Great presentation. Thank you.
Nelson.
Other questions?
I haven't even looked in the chat. I don't know if there's any in the chat. Sorry, I'm not very good.
Lots of praise, but I do have a question. You talked about how the engineered B cells can be trafficked through the lymph system and so forth. Can they make it to the brain?
That's a really good question. We don't know. We haven't looked, but we also are funded on a different grant where rather than thinking about trafficking the B cells to the brain, we're using some of because we've got so much flexibility to make custom antibodies, we're thinking about making antibodies that will specifically cross the blood-brain barrier more efficiently than normal antibodies. So we're adding like an extra capability, so an anti-HIV antibody that also will cross the blood-brain barrier. So that's another kind of idea that the guys in my lab had. They're like, not, exactly like you. They're like, why don't we do this? I'm like, oh, okay, that's a good idea. And we have a lot of expertise using humanized mouse models that have HIV infection in the brain, and the special cells in the brain called microglia that get infected. So in theory, we have the tools in place where we could analyze, do our antibodies get into the brain, and do they impact HIV infection in the brain, at least in the early stages with a mouse model. As you know, when you start thinking about doing anything in the brain, it's massively more complicated. So you have to start with mice.
Yeah, wonderful talk, Paula. That you were talking about manipulating the FC portion, right?
Yeah.
So this would also recruit the NK cells. Is that something that you're looking for? Okay.
Exactly. So in RID-HIV, we have another project with Jeff Miller where he's all about taking advantage of NK cells and recruiting them to HIV infected cells by making these sort of bridged antibodies that on the one hand bind to an HIV infected cell, on the other hand bind to the NK cells. So having NK cells do their job, but on steroids. And because he's part of our collaboratory, and when I started talking to him and reading his work, I thought, oh God, I never thought about that. That's a really good idea. And so now we're thinking, can we do what he's doing, but do it from the point of view of engineering the antibodies to kind of act as the bridge anyway through the FC stalk. And I've also been talking to Galit Alter, who's on our scientific advisory board, and she's on I4C. So it's all friends and family. And she's an expert on doing this as well. So I think there's, we're the engineers. So I'm like, if we can show people that we can indeed provide this capability to engineer the stalk, people can tell us what we should be putting in there. I don't need to be an expert on that as well. I can call people who are experts on that. So I see lots for collaboration there.
I mean, fantastic talk, Paula. I should say that, you were able to dumb it down, because most of the people don't understand the gene.
I don't either.
So it's complicated, when you broke it down like the
pieces. It's very complicated.
Fantastic. It's recorded, and I'm glad, Jeff would be able to share, and I can help other people understand this thing. Wonderful.
Okay, right. Well, it's at the level I think about it. So I don't think of it dumbed down. I'm just like, this is all you need to know.
No, I think it made it very simple for me. Anytime the Cas9 guide and all that, I try to read. I understand then when I read, but after some time, would I be able to explain to somebody? Then that's
all right.
When
I'm going to test you afterwards. Okay. What can I say? I am a professor. I like to teach.
We didn't warn the audience about the test that comes after. And then somebody in the chat asked what could go wrong. And one specific question I had about that, if you're using, say, llama genes, is there a danger of the human immune system rejecting these?
Absolutely, absolutely. Yeah. So that llama gene, the J3 that we use, that was kind of as proof of principle. But in another collaboration with I4C, one of the things their team are doing is they've humanized J3 for us. So they've made it look more like a human antibody, which should get away from that. So that's one strategy. Of course, the other strategy is we just use things that came from human cells, like CD4. That's, we won't make antibodies against that. But the safety concern I'm most concerned about, Jeff, as a gene editor, is I'm going into B cells and I'm making DNA breaks, very controlled and specific, but I'm still making a DNA break in a B cell. And B cells are one of the cells in the body we know can undergo translocations and chromosomes as a result of breaks moving around. And sometimes that can result in leukemias and lymphomas. So I'm very keen to move beyond what we're doing with CRISPR-Cas9 DNA breaks and instead start to use some of these emerging technologies which allow us to do the exact same gene editing, but without making a DNA break. So there's a lot of excitement and movement in the gene editing field. So again, we started off with vanilla CRISPR-Cas9 and our llama antibody as proof of principle. But boy, there's new things coming down the pipe that we're going to switch to, because I think they're likely to have a much greater safety profile. And and I recognize always that, developing a novel treatment for HIV, while it's super exciting to think about that, you guys don't need any risk in your lives. You guys are doing fine. So the barrier to be safe here is super high as far as I'm concerned.
Yeah, excellent. Thank you. Any other questions for Dr. Cannon?
I would like to ask Paula. I guess you're going into mature B cells. Is that right? The idea is to pull out mature B cells and.
Yes, and at the moment, we're just pulling B cells out of the blood, which is a combination of naive B cells that have never met their antigen and memory cells that have been boosted. So we can alter both of those. But in theory,
right? But
you're going to
not pre-B cells is my point. And so all of these will have their own endogenous receptors, and you're not knocking those out.
No.
So in theory, you could also be selecting for. Antibodies that, you already have a negative, you're not necessarily autoantibodies, but you could be selecting for antibodies that, kind of come along for the ride. Is there any concern about that?
No, not so much, because again, any B cell that's kind of floating around in the body making an antibody, even if it's not churning it out, it's already gone through this kind of process of selection against self. So in theory, it's a safe antibody. So if we also activate some production of antibodies, remember, we're going in and destroying the heavy chain. So only a subset of our B cells would still make their original antibody. And our expectation is it'll just be innocuous. It'll be like being vaccinated against a virus that you never meet. All the time we churn out antibodies that don't do anything as a result of vaccination. So we don't think it will be a problem. However, we also do have the capability to engineer pre-B cells, the hematopoietic stem cells, which is what my lab used to work on all the time. And we could do that strategy as well. It's a very similar approach. We're focusing on the B cells because I love the fact you can just, in theory, we could take, 50 mils of somebody's blood and get enough B cells to work on and then pop them back in and then let vaccination and boosting do the amplification for us in vivo. So that's the big appeal for me of going into B cells. But we could, if that proved problematic, we could go many steps back as well.
Yeah, that was kind of my question. Can you do it in pre-B cells?
Yeah,
you already have some experience doing that. Yeah.
No, we don't. But I can draw the cartoon, and in the cartoon it works. So I think it should work as well. Yeah.
So just to clarify for kind of lay audiences, so right now you're looking at this is individualized gene therapy where you're taking somebody's own blood, their own B cells, and reinjecting them. What's kind of the next step to making kind of a one-size-fits-all
Yeah, no, that's a great question. So, so of course, two things that we're also thinking about. One is how to do this in vivo, how to inject somebody with the gene editing machinery that will go to a few B cells in their body and do this for us, so we don't even have to, take anything out, as a way to make this more widely available. And that's paralleling, I think, efforts in the gene therapy field in general, where people are really excited about the idea now of putting effort into in vivo gene therapy. And then, sorry, what was the other thing I was going to say? I've forgotten. Oh, yes. And then we also have some ideas about how to make a more sort of off-the-shelf B cell that could be given to everybody without the normal sort of tissue compatibility things. And again, this is paralleling what people are doing with CAR T cells, where there's a move to thinking about making off-the-shelf CAR T cells. So all these things are possible. And I love the fact that, we get to plug into so many parallel technologies that are being developed, with our focus on engineered B cells, heavy chain only antibodies, HIV, but there's a lot of things that we can collaborate with and bring to bear on this task that, my little lab alone couldn't do. And I think I saw a question asking about whether or not Big Pharma's involved. Yes. They have not yet made me the right offer for our technology, but we're certainly talking to a number of sort of medium-sized biotech companies as well. So I think, and there are a few biotech companies being developed. So in the same way that I think, in particular, Novartis were out there ahead with taking advantage, I think, very early on, and I give them credit for this, for the CAR T cell technology, I would expect that this field will also become attractive to not Big Pharma so much as Big Pharma that is already invested in and sees the promise of cell and gene therapies. And, it's the medicine of the future. You don't want to put chemicals in your body. You just want like a nice, perfectly produced B cell doing what B cells do naturally, but, helping you to fight HIV. Anyway, sorry, that was a stretch. I know.
Well, I did see Gilead's logo on your slides, so they're in the game, and what they always do is find somebody who's come up with something promising, and then they swallow them up to the tune of billions of dollars. And
if they want to throw money at me, that's fine as well. But I think where we'll go probably with the technology first is plug into, either set up our own company. I'm talking to some VCs, but I'm also interested in plugging into some established B cell companies, because there's a few that have sort of figured out how to, all the sort of culturing and how to grow and safely monitor B cells, but haven't really thought about the gene editing part. So that seems like a nice marriage to me, and an efficient way to get this technology moving. So I'm, that's the other thing I do. I'm pitching this to some of those companies, and we'll see which ones bite first. Excellent.
Well, good luck with that. Well, we're past our hours, so thank you again for this excellent talk. This will be archived on the RID-HIV website, and I put the link in the chat there, and also on the Heart PS YouTube channel as well. So, and next quarter, stay tuned. We'll have another excellent seminar. So thank you again, Dr. Cannon, and to everyone for joining.
Welcome.
Thanks a lot. Thank you.
Thank you.
Bye. Bye.